What up? It's your boy Nickel. Sci-Fam, let's do this. This one's a twofer. Oh, you want to know what a twofer is? What that means is we got 2 note pages for 1 video note page. Video notes video. So today we're going to learn how DNA codes for our traits. So go ahead and instead of codes for, I just put the arrow there. Make sure you put that title at the top of your page. You can kind of see that after we finish talking about an mRNA strand, we'll start page 2, and I'll have a notation here in the video. So let's begin. So to begin with here, a DNA strand, as we've learned, is a double-stranded molecule that creates a double helix. And so when I draw it, I like to draw the 2 lines kind of overlapping each other and then draw like the ladder rungs that connect the 2 lines to each other. The ladder rungs represent the bases, those 4 bases that are part of DNA. Remember, it's A for adenine, T for thymine, C for cytosine, and G for guanine. So it's one of those 4 letters will be used on each strand of the DNA, and the line represents those 2 connecting each other. So if I were to build a DNA sequence, if I was to rip those 2 strands apart from each other, I could make a DNA strand that was like, let's say we have adenine, thymine, cytosine, cytosine, adenine, guanine. If that was the DNA sequence on this top strand, that means the bottom strand would have to be complementary, meaning that it has to be similar, or it has to be the opposite pair of the top letter. So A pairs with T, T pairs with A, C pairs with G, C would pair with G, A pairs with T, and then G is going to pair with C. So there we have our DNA sequence on each strand. And so if we ever only know 1 strand of DNA, we can always figure out the second strand because it's a complementary sequence. So we would say that the DNA sequence is used as a tool. It's like the instruction manual for our traits. And so when we talk about the DNA sequence, we would say that it's actually going to be used to create a messenger. And this messenger we call RNA. It's similar to DNA, but it's a little bit different. We're going to talk about how RNA is different than DNA. So DNA is like the instruction manual to create a messenger RNA. And so in order to abbreviate that, we put in lowercase m, then capital RNA. So the DNA sequence is used to create a messenger RNA strand to send the information about how to do our traits. Send the information for the trait out of the nucleus. Okay, that was a little bit clunky there. But essentially, the DNA is used to create a messenger. The messenger is called RNA, so we call it mRNA. That mRNA strand can leave the nucleus and go out into the cytoplasm and go find the ribosomes, which will make us our proteins. So the DNA is not allowed to leave the nucleus. It can't leave the nucleus. We don't want it to in case it never comes back, or if it gets destroyed, that would be tragic. So instead, we use the DNA to create a messenger molecule called mRNA. Now, if we look at an RNA strand also has 4 base pairs. It's very, very similar to DNA, but the 4 base pairs here are adenine, which we abbreviate with A. The new one is uracil, which we abbreviate with a U. Guanine is still one of the bases that's similar to DNA, so guanine, and then finally cytosine, and cytosine abbreviated with a C. So when we pair these up, because we have a new letter here, uracil actually just replaces the thymine. So adenine is still going to pair, not with thymine this time, but with the replacement with uracil. So A and U pair together, and then the G and the C are still going to pair together. So whenever we have an RNA molecule is going to use this configuration for pairing, or to at least design the RNA strand. So in order to explain what I mean here, if we have this top sequence, since it has a T thymine in it, that means it's going to be DNA, because RNA doesn't have thymine in it. So we know that if this is our DNA sequence right here, we can then build our RNA sequence. So A is going to create a U on the RNA strand. The T is going to create an A, or an adenine. C is going to create a guanine. Cytosine is going to create a guanine. Adenine is going to create a, uracil. Guanine is going to create a, cytosine. So we use the DNA sequence to create an RNA sequence. The RNA sequence can now leave the nucleus, and it's got this secret code from the DNA, and so it's going to leave the nucleus out into the cytoplasm in search of the ribosome, because even though the RNA has a sequence, the ribosome is going to translate that sequence into something that's useful. So a strand of mRNA, it's got a secret code here, and it has to make sure that it's protected and doesn't mess anything up. So in order to protect it, a strand of mRNA protects the RNA sequence by adding a cap to one end and a tail to the other end. So up here at the top, it puts something that we call a 5' cap. And then you have your RNA sequence. So like, for example, if we look at above, it would be in the RNA sequence, we would have U, A, G, G, U, C, which is that secret message. We want to protect that secret message, so we'll put that secret message right after we put a cap on the top. We call that a 5' cap, which would just be some other base pairs there that would create the cap. It would then put the RNA sequence in there to protect it, and it would follow it with a bunch of A's. And if there's a lot of them, we would call that poly. So we call this a poly-A tail. So we have a 5' cap, an RNA sequence, and then the poly-A tail. The RNA sequence is what has the information that needs to be translated. Now, something that's different about RNA is that it's actually just a single strand. So we don't have a double-stranded molecule like DNA. RNA is created from one of the strands of DNA. It's transcribed into RNA. RNA then takes that message to be translated by the ribosomes. And now page 2.